Hello students. Today we will start with the next enteric pathogen, that is Salmonella. Our objectives for lecture will be classification and biochemical characters, antigenic structure and viability characteristics of Salmonella. Okay. Now, Salmonella, they are the widespread pathogens of vertebrates, including human being. Okay. Salmonella typhi is the most important member of the genus Salmonella that causes typhoid fever. Ebert, in 1880, he first observed the typhoid bacillus in mesenteric lymph nodes and spleen in fatal cases of typhoid fever. Okay? And Gafke, in 1884, he successfully isolated the organism. It was then called Ebert-Gafke bacillus or Eberthella typhi. Okay. Next, Salmon and Smith in 1885 isolated the American hog cholera bacillus. It is S. choleriasis. And from the name of the first author, the term Salmonella is derived. Understood? Now let's see the characteristics. Salmonella, they are facultatively anaerobic, gram-negative bacilli and primarily intestinal parasites of vertebrates. They are enteric, okay? They do not ferment lactose on McConkey agar. Produce gas when fermenting glucose and produce H2S from thiosulfate. Understood? They are pathogenic for many species of animals and produce enteritis and typhoid-like disease. S. typhi and S. paratyphi A differ from other species in that man is the only natural host. Understood? Next, Salmonella species, they are non-spore forming. They are motile enterobacteria with cell diameters between 0.7 to 1 and 1.5 micrometer lengths from 2 to 5 micrometers and peritrichous flagella all around the cell body. Okay, understood? They are chemotrophs, obtaining their energy from oxidation and reduction reactions using organic sources. They are also facultative anaerobes, as I told you. Okay? Capable of generating ATP with oxygen aerobically when it is available, or when oxygen is not available using other electron acceptors or fermentation anaerobically. Understood? Now, sets of antigen. Somatic flagellar virulence, OHVI. Somatic antigen, they contains long chain of lipopolysaccharides, LPS. Flagellar H antigen, strongly immunogenic and induces antibody formation rapidly, and in high titers following infection or immunization, they are dual in nature. Virulence agent act as virulence factor and protects the bacilli against phagocytosis and activity of complement. Understood? Its antigenic structure. This is the structure. Okay? See here. Salmonella classified on the basis of the serotype, either typhoidal, either non-typhoidal. Typhoidal cases, it causes typhoid fever and paratyphoid fever. Example, Salmonella typhi and paratyphi A, B, C. In non-typhoidal conditions, it causes gastroenteritis and food poisoning. Example, all other Salmonella serotypes. Understood? The mainly causing typhoid fever and paratyphoid fever are Salmonella typhi and paratyphi. Understood? Rest, they are non-typhoidal, causing gastroenteritis and food poisoning. Understood? This is its classification. Biochemical characteristics. Now here you can see different tests are done. Serotypes, typhi, paratyphi, with respect to typhi and paratyphi. Okay? Now here you can see H2S, TSI, citrate, lysine, ornithine, dulcitol, rhamnose, indole, methylred, VP. Okay, I told you MVIC is done whenever some coliform group is present. So here you can see citrate, indole, methylred, VP. Got it? Now typhi and paratyphi A. What all reactions they give to respective chemicals? For TSI, triple sugar ion, most serotypes are alkaline. Typhi is alkaline, paratyphi is also alkaline. For H2S, typhi is weak positive, paratyphi is negative. Citrate, serotypes positive, typhi is negative, paratyphi negative. Lysine, serotypes positive, typhi positive, paratyphi negative. Ornithine, serotypes positive, typhi negative, paratyphi positive. Okay, so this way you can see the rest. So what will be the MVIC for typhi? Negative, positive, negative, negative. Understood? For paratyphi, negative, positive, negative, negative. Understood? Okay, clear? And for serotypes, it will be negative, positive, negative, positive. Understood? Next, viability characters. Now, Salmonella species, they lead predominantly host associated lifestyles. Okay, they require host every time. But the bacteria, they were found to be able to persist in bathroom settings for weeks following contaminations and were frequently isolated from water sources, which act as bacterial reservoirs. You know that. And may help to facilitate transmission between hosts. Okay, means it was thought that Salmonella is only found in a particular host, but no, it was not the condition. They were also found on bathroom settings, like for week if the water is clogged somewhere, sewage. Salmonella was found over there also. Salmonella was also isolated from some water sources. Understood? Salmonella is notorious for its ability to survive desiccation, means in dry places also it can survive. Understood? And can persist for years in dry environments and foods. Got it? Salmonella species can be found in digestive tracts of humans and animals, especially reptiles. Salmonella on the skin of reptiles or amphibians can be passed to people who I'm sorry, there's a mistake over here in the writing, who take care of those animals, or who veterinary people who handle them. Okay, food and water can also be contaminated with the bacteria if they come in contact with the feces of infected people or animals. Okay, understood? This is its viability character. Now in the next lecture, we will study its pathogenesis, symptoms, laboratory diagnosis, treatment, prophylaxis. Okay, if you have any doubts, do ask me.